Evening, everyone. And to the people who are joining from the US, a very good morning. Um, just a few announcements before we start. In case there are any questions, there is a Q&A section at the bottom. So please send your question and answers there. If there is any other chat, then you can use the chat button. In the last 10 minutes, we will take up your questions. So whatever comes to your mind, please go ahead and type in there. So I'm Dr. Archana Beri. I am your host today and also the moderator for the webinar. We have Dr. Richard Bonneau with us today as our speaker. I would like to take a few minutes to introduce him. Dr. Richard Bonneau is a computational biologist and data scientist whose primary research is in learning networks from genomics data, predicting and designing proteins and peptidomimetic structures. He has diverged from applying data science to applying data science to social networks. He leads the systems biology group at Flatiron Institute, Simons Foundation, New York. Dr. Bonneau is currently the professor at Department of Biology and also director of the Centers for Genomics and Systems Biology at New York University. A co-director and a co-founder of Center for Social Media and Political Participation Labs, New York. He is talking to us on the recent advances in machine learning approaches at the intersection of protein sequencing or language models and the use of these models in settings that incorporate protein three-dimensional structures. He will discuss his work on Deep AI, a method for using language and graph convolutional neural network models to predict design protein structure and function. He will also discuss new methods for using these models to find similar proteins and implications for scale. Lastly, he will discuss how these ML models can be integrated with generative approaches like Rosetta to build design and prediction pipelines that scale to metagenomics and beyond. I am extremely pleased to hand it over and introduce Dr. Bonneau today. Dr. Bonneau, please take over.
Thank you. Thanks for the invitation. And I see that we have a great number of panelists on the and attendees. So thanks to everyone who's attending as well for giving me some time. I will switch over to my talk here. All right, so today I'm going to give you a talk that has maybe a few too many parts in it. And so I apologize in advance if I try to fit too much in. But in the end, what I really want to do is dive deeply into some machine learning methods that aim at, given a protein sequence and structure, predict its function. And once I do that, I want to pull back and say, well, if we can do that, then there are a number of other things that we can do. And so I want to give you a small window into how we use similar methods to just find similar proteins in the first place, so the alignment and homolog detection problem. And then the third thing that I want to maybe give you just a little hint on is some work done by Vikram Mulligan in my lab, who's working on accelerating design, protein design using machine learning methods. And so all of this work, I will be talking a lot about the work that comes from these people in particular. There are a great number of people in the lab and at the Flatiron, but these are the people who contributed most to the core of what I'll talk about today. So first of all, Kyung-Young Cho is a machine learning specialist and professor at NYU. He actually is famous for developing things like the attention mechanism and recursive algorithms that are the state of the art right now in machine translation. So we're very lucky to have him to work with. Julia Kohler-Lehmann, she is a research scientist at Flatiron, along with Vlad, Vikram, and Jamie. She develops Rosetta and is one of the core developers of the structure modeling code that we'll talk about. Vlad is a computer science PhD turned Flatiron fellow. His background is in non-negative matrix factorization and other things like that. And he now works almost exclusively on protein structure function machine learning. Vikram is someone I'm very lucky to have in the laboratory. He right now is very focused on protein design, but he has learned a huge amount of machine learning and is applying that to accelerating Rosetta. So tensor versions of Rosetta and all of these sorts of things. Jamie actually studies the microbiome. And here he's interested in working with the rest of these crazy people because he wants to use these structure function tools to annotate the microbiome and the huge diversity of unknown proteins that live in bacteria that live in us. So he's got a lot of motivation to use structure and structure sequence machine learning methods to annotate these huge undiscovered territories that are hiding in the biodiversity in microbial ecosystems, including the ones that live on us. Daniel Berenberg is, again, another person we're very lucky to have on the team. He is now a PhD student at the Center for Data Science and just started, but he worked with us as an intern for about a year before that. And so he's also contributed a lot to the papers that I'll talk about today. Whoops, that's not how I advance the slides, sorry. And again, if you have questions, stop me throughout, I guess through the chat, and maybe one of the moderators or other panelists can interrupt me. All right, so we'll, since we're all from many different backgrounds, so one of the beautiful things about biology is that it's so hard that you need people who have just about every kind of undergraduate degree to get together and study it if we have any chance of cracking the puzzle. But this is what we often start with. So we start with the organism. Sometimes we start with an ecosystem. Sometimes we start with a subsystem we care about. But for us these days, after the molecular biology revolution, what we many times start with are sequences that are just strings of amino acids or bases, and we just have no idea what they do. And so that's why I've put in the top here, this is a FASTA entry for P53, which is a very widely studied protein, and I've just replaced its annotation line with all of these question marks to pretend like I don't know what this protein does. Now, of course, that protein that I just showed you is P53, and this is a stereo diagram. So if you have had just the right amount of tea and coffee, you can cross your eyes and you can see this structure in 3 dimensions. And of course, the sequence folds into this protein, and here we can see it binding DNA, carrying out its function. So for many proteins and many genes that make protein products, we have a huge amount of information about them. But for many proteins, at least half, all we know are question mark, question mark, question mark. All we know are the sequence, where it starts, and maybe what organism it's in. For microbiome proteins, we might not even know the organism that the protein is from. It might just be a fragment of DNA from a patient or an environmental location. We still very much might want to know what that protein does, but all we've got is the sequence. So this is where we start. In some special cases, we have structure prediction. Key to this talk is that even when we don't have structure, we can predict it. And we'll work through that together, get to that point together. Again, though, before I start telling you how I'm going to predict function, I want to spend a little bit more time telling you why we need to predict function. And so this is a picture from a paper by Jill Banfield, who's a very famous microbiome environmental ecology genomics enabled microbiologist. She does a lot of great work. And so if you don't know of Jill Banfield's work, I would encourage you to look it up, and there's all kinds of things here. So this is a tree of life that is sort of impressing upon us that the top part here, this top part here is actually fairly large compared to the biodiversity of the known eukaryotes. Now, there are lots of efforts to sequence many new organisms, but even with the organisms we see today, and not the 10-fold or 100-fold more that we'll have tomorrow, there's huge amounts of unexplored biodiversity, organisms whose role in their ecosystems we barely understand, we're just beginning to understand. Organisms that have been sequenced via environmental sequencing but are otherwise totally uncharacterized. And each of them has a large proteome filled to the brim with millions and millions of unknown proteins. So there's a lot of biodiversity to explore. Now, whether we see this as a challenge for keeping the library that we're building organized, sort of bioinformatics challenge, whether we see this as a mining biodiversity, like an industrialist challenge, we want to find enzymes that do things, or whether we see this as just the basic science of understanding life, we have quite a challenge here, no matter what our perspective is. I see someone's raised a hand here. Do you want to, oh, what have I done? I'm not quite sure how to see who raised their hand without stopping this.
Dr. Richard, I'll note down the questions and we can take it in the end.
Oh, okay, great, okay. All right, so that is the tree of life, and that was meant to just scare you into thinking, holy cow, wow, there are so many proteins, what are we gonna do? Okay, so feel the motivation, and now we'll move on to some of the details. So now we're getting into some of the details. If we want to predict the function of proteins, hopefully I've done a little teeny bit to convince you that it's an important task. Now we have to actually talk about what is the task of predicting a protein's function. Again, with three-dimensional structure, I can show you a picture and say we write down the coordinates in a rotation invariant frame, but fine. We write down the coordinates of the molecule. And each atom in the molecule gets an X and a Y and a Z. With protein function, it's a little bit different. Here, the coordinate that we're writing things down in is a tree called the gene ontology tree. There are also other trees and other systems. The enzyme classification system is one, and there are many others. There are a couple others. But this is the one that we'll focus on today, the gene ontology tree. And so the idea is we have very general terms that apply to lots and lots of the genome at the top. And then we make these labels more and more specific as we descend the tree. And so here we have an entry for a protein involved in the immune system. And so at its most specific, we have regulation of cytokine activity. Eventually we get up to regulation via protein binding, repression and activation, some very general concepts. But at the bottom, we know that it binds collagen, that it binds, that it is a cytokine, and it is the cytokine that binds a receptor and not the receptor that binds the cytokine. So we're starting to see the picture. This particular annotation descends further until we all but name the protein. So the goal here is to predict the most specific terms we can, but also to properly predict these general classifications, which might be useful too. So for example, transcription factor is a very general term, but we might want to design a screen where we perturb all the active transcription factors. So the big buckets are useful when we're designing studies and genomic large-scale screens. The smaller, more specific buckets at the bottom of this tree are very important, because those are ultimately the functions that biologists want to test in the lab and want to understand when they put together systems views of cells. Unfortunately, again, I'm sort of beating the drum that this is an important problem. Unfortunately, there are actually a lack of experimentally derived annotations for most organisms. So if we look at human, there are 500,000 annotations, and less than half of them are experimental. Many of them are mapped, and therefore mapped from other organisms or other contexts. And so there is a lot of need to reconcile this information and fill it in. The other thing to point out is that there are many tens of thousands of human genes. And so the fact that there are 500K annotations, that doesn't mean that everything's annotated, because human proteins have on average 2.5 domains. They're big proteins, so they have multiple functions often. And this tree has many terms on it. And so included in that 500K are all of the instances of these really general top terms. And so in spite of the fact that we have that large number of annotations, most parts, about half of the domains in human remain unannotated. And that's one of our best studied organisms. When we get to things that aren't amongst these well-studied organisms, the story gets more stark, more sparse. So here are just some papers that I would point people to that are at this point a bit on the older side. They're about 4 years old, 5 years old, but these are some nice papers where they have tried to test methods for protein function prediction. This one on the left, where Jiang is the first author, this is the production from the CAFA effort. And so this is an effort to once every year or two, release a bunch of sequences and say, we expect annotations for these sequences or some of these sequences. We make predictions for all of the sequences, and then we judge the predictive performance of methods on the subset of sequences that were annotated in between, in the 6-month period between when the competition collects the predictions, and 6 months later, we evaluate the predictions. And this is run by a number of people, including Ido Friedberg. Hold on a second. Daddy's giving a lecture, so just get ready for school, okay? All right, sorry, small interlude. It's morning here, so the kids are waking up for school. This second paper here is earlier still, and here they took the early mouse genome and got together many different people who work on function prediction methods, and they were able to make some progress in deciding which methods work best on this large, complex mammalian genome. So these are two works that I would point you to. Of course, CAFA has published since this paper, and everyone on the mouse func paper has done a lot of work since then as well, but I like these two old papers. Okay, so the last piece of the puzzle before we're ready to start actually talking about what we do in my laboratory is go upstairs and get ready for school. Okay. The last thing that we need to talk about are the general types of features that we might use to predict protein function. And of course, if you're not from a machine learning background, when I say features, I mean the variables, the qualities, the extracted variables or extracted qualities of the genes that we might use as the basis for predictions, the foundation for prediction. And so here, the features might be, what is the sequence of the gene that we're trying to predict the function of? And I might look at here, the protein sequence. I might also derive some information from looking at the protein and the origin DNA sequence, who knows? Here, I'm also showing you not just the sequence, but a derived product of multiple sequence alignments. So at the top is the protein we're trying to predict, and then lined up are the many similar sequences that we found in other organisms. We might look at the three-dimensional structure of the protein. So we might look at sites on the surface. We might look at its stability. We might look at all sorts of derived features from the three-dimensional structure. And those would be features, including the main ones I'll talk about today. And then another really strong set of features for predicting protein function are network features. And so these are features aimed at what the neighbors of a given protein are. So we might sit on a single node for which we have the sequence and structure, but we will also know things like, is this protein in an operon with other proteins? Is it interacting with other proteins in metabolic pathways, or is it interacting with them through physical interactions, protein interfaces? So these are the three main types of sequences. We've done a lot of work in the laboratory to extract features automatically from collections of networks. And this is work by Meet Barreau and Vlad Grigorovich. And Meet has just put together, Meet is a PhD student at the Center for Data Science and is really working very closely with Vlad, is very deep into the machine learning literature and applying it to biology. And his last work is now on bioRxiv, so check it out. It's called NetQuilt, where he actually uses deep learning to integrate species across multiple networks and finds ways to make predictions, even when organisms don't have a very large number of annotated edges. So this is really nice work. this isn't what I'm going to talk about today, but I just wanted to pause here and show you this paper in case you're more interested in networks than you are in three-dimensional structure. So, and there's going to be a lot more to come from this brilliant young PhD student. So we'll also maybe put a note in your calendar to Google these names about a year from now, because I think Meet is working on something else pretty cool. All right, so that's my little advert. Check out the guy's paper. Here, though, I'm going to switch back to sequence and structure. What do we do with sequence and structure? And I'm going to take a little detour into the algorithm, and then I'll show you in great detail what the algorithm does. So first of all, let's think a little bit about how machine learning methods process text, or process images, or process many of the things that aren't biological. And the first thing to think of is just that when machine learning methods extract features from images or from text, they do so using the native coordinate of the image, right? So if we talk about a convolutional neural network, a CNN, what it's actually doing, and this is all you have to think about for now, is the perceptive field, is it is looking at that image one window at a time. It's then summing up what happens in a reduced dimensional matrix of features, and then those features are convolved and convolved and convolved. But the initial thing that happens is that the image is scanned essentially by the first layer of this convolutional neural network. And then through convolutions of this green top matrix, we can get all kinds of relationships between distal parts of the image. But the model has to learn that. Inference is about different parts of the image coming together through convolutions, has to be learned by the weights of the model. And so with sufficient training data, we'll learn the rules to put different parts of images together. But there's another approach, which is for things that aren't easily embedded in these 2D spaces. So many things in the world that we care about, instead of represented as being placed in a three-dimensional coordinate or a series of spoken phenoms, a 1D coordinate, many of the things that we care about are actually best represented by graphs. And so we might think of a social network where who you interact with online is represented by edges. We also might think of a protein as a graph. And so here, what I've done is I've taken any two C alpha atoms. And so the C alpha, again, is the atom that the side chain branches off of. So it's a very important atom in a protein polymer, in a long protein polymer. I've taken those C alphas and I've drawn an edge between them here for just for illustration, if those two C alphas are less than 10 angstroms apart. Okay, and so then we get this dense graph, but it has these clear 3 clusters that represent the 3 domains of this folded protein. And so if we represent a protein by its graph, then we posit that if instead of doing convolutions along the primary sequence of the protein, if we do convolutions that pay attention to this graph, then we could get the convolutional neural network downstream to see clusters, groups, neighborhoods on this network. And those neighborhoods would correspond to patches on the structure, structures in the core, and various other punctate or densely packed areas on the protein. And so by traversing the graph, the CNN, or the graph convolutional neural network, properly put together, can actually look at the protein structure in what's perhaps a more natural framework, a more natural frame. And so this is how it works. At the very first iteration, we decorate the protein, each amino acid, with its features. And maybe the features are, what is your amino acid? That would be called a one-hot encoding of the protein. We can also do things like run so-called language models to extract more complex features and then decorate the primary sequence with more complex features. We can put whatever features we want. And then what we do is we diffuse those features essentially over the neural network model, so that by the time we get to maybe say 3 layers of graph convolution, every given residue has a combination or a pooling of some sort of its features with its neighbors in the graph. Okay, and I am gonna just show you really quickly. Now, all of this stuff is because this is a lecture, right? And we don't have 4 hours. We don't get to break and go to lunch and then come back and ask more questions in front of the whiteboard. It's going to be a little tough to get all these details, but they're all in this paper here. And so this is a paper, again, that involves people at MIT, New Zealand, Poland, New York. There's people all over the world involved in this paper. And this paper is where you find all the details, including setting up the training set, training the model, links to the code, and all this kind of stuff. So, and again, this is something that I'm willing to share. It's already on the bioRxiv, so it's already freely available. All right, so with this graph convolutional neural network, that's the framework we're going to use to integrate sequence and structure. We still have to address the elephant in the room. We have to address the large, really potentially quite critical problem that not everything has a structure. Genomes and proteomes don't come with three-dimensional structure, they come with just sequence. And so we have to, in some manner, in some computationally efficient manner, right? Because we have millions of proteins, so we can't take all day, can't take all month on the cluster for each protein. We have to find a very fast, very efficient way of predicting the three-dimensional structure. And for that, we use Rosetta, which can do many things. It can do protein design, it can address issues modeling membrane proteins, which is Julia's specialty. It's widely used in industry to hyperstabilize and modify antibodies for drugs, all sorts of things. The part that we care about is the part that Doug is most familiar with, or one of the parts that Doug is most familiar with, which is the prediction of protein structure. And so here we derive relationships, coevolutionary relationships between different positions in the sequence, and we use those to generate constraints. We feed those to Rosetta, and then Rosetta predicts the protein structure. And to get the computational power to do that, we've teamed up with IBM. And so we're very thankful to IBM and the World Community Grid for that. Here there are many methods, including methods from Debbie Mark's group, methods from the Baker Lab, methods from the Jones Lab. And so we actually, in this particular case, we use methods that come from David Jones and his group, and Tomasz here is the first author on some of that stuff, and you'll see him later. And if you would like to help us out and you have some extra electricity, and probably nobody does, but if you have idle computers, one thing you can do is you can connect your PC or your phone or your laptop of any kind to wcgrid.org. And by volunteering in that way, you'll be folding proteins for the project when you're not using your computer. So this is grid computing. And so with this project, we get many millions of CPU hours every day. And the structure predictions that we produce are very accurate in this particular case, because we actually have very careful quality control connected to Rosetta, because we have enough computational power to fold every structure and really learn when the method is working and not, and correct many of those things. So this is a case where we had predicted contacts from coevolution in this top purple box, and you see there's a sparse but fairly accurate subset of contacts. This is the true answer from the folded, densely packed folded structure. And when Rosetta reconciles those contacts and produces a full proper three-dimensional structure, we see that Rosetta's predicted contacts, which again are going to be the key input to our machine learning method, those contacts are fairly accurately predicted. And of course, in addition to predicting function, we're also exploring lots and lots of new folds as we put millions of proteins through this. And we now have a database that we estimate has as much structural information as the PDB itself. So, and again, here is where you can go to actually learn about all of these things in detail. And details are important. Here's a nice schematic of the final method that we'll use for function prediction. So I've told you a little bit about what a graph convolution is. I've told you that we're going to go completely nuts and try to predict the structure of everything. You know, that maybe that's a good idea. Maybe that's a little bit over the top, but that's what we've already done. So the input will be a sequence or a structure. If the sequence doesn't have a structure, we will predict one. This will give us a contact map, which will represent as a network of adjacency or a neighborhood of each residue. So every residue will be surrounded in the network by the proteins that it contacts. Okay, the sequence will be run through a long short-term memory language model, and so this is one of many different types of machine learning methods, including Roberta, including attention mechanisms. There's many things that we've tried here, and this is the one. It's a bit older, but it does seem to be the proper architecture for proteins in this particular case. This model, essentially, if you hide part of a sequence, it can predict the part that you hid. So this is a self-supervised method that aims to encapsulate the hidden language of protein sequences. The output for this is that now we get a vector of the derived features for each position in the sequence. So instead of the primary sequence, we now have a vector from the language model. That then is used to decorate the nodes in the graph. And those features, these language model features, are diffused through the protein structure graph by this graph convolutional neural network. That gives us our final feature vector, which then goes through the remaining dense perceptron layers to predict the thousands of potential function labels at the end. So that is the architecture. And again, that's a lot of details. All of those details are out right now. So, okay, so the first thing we have to do is figure out if this works. So there's a bunch of different things that are advantageous about this method. But if it doesn't outperform other methods, then we don't want to dig into the details of methods that don't work. And so here we have broken down by, on this x-axis here, I have different bins. And what I'm binning the test set by is the maximum % sequence identity to the training set, right? So because we have evolution, we can do most of what we do in biology. But sometimes when we test methods, evolutionary relationships between proteins fool us into thinking we're doing better than we think, right? We might predict something for a protein and we think that we've solved the problem. Oh, this new protein, we predicted its protein function, but we might be fooling ourselves because that protein might be very strongly related evolutionarily to a protein that's in our training set, and we haven't solved anything. We've just memorized one protein. Okay, so what we do is we actually decrease and decrease the size of the training set, and we do that by removing similar sequences, so that essentially we end up with training sets and test sets that don't have sequence relationships. And so we find that as we exclude sequences from the database so that homologs are standing by themselves, we decrease the predictive performance of FunFAMs, which is a sequence homology domain-based sequence method, DeepGo, which is a deep convolutional neural network trained on sequence, and DeepFRI, which is our method, which is a sequence structure combination. We find that our method actually falls off slower than the other methods, and in each bin outperforms other methods. So we think that we're not just memorizing sequence patterns, but we're actually figuring out what those sequence structure patterns are and generalizing much better. But either way, we perform better than other methods that we can test, including state-of-the-art methods like DeepGo that use a lot of the same deep learning technology that we're using. Another question we have is how well can we tolerate error in the predicted structures? And so here what I'm showing is a precision recall curve that's showing how accurate are function predictions, and then how many on the x-axis, the recall, what fraction of the known functions have we predicted? And so you can see that our precision up until about 40% of all known functions is fairly high. We have above 95% accuracy even as we get to predicting, half of the known things in the database. And then there are some functions down at the lower right here that resist our efforts to predict them no matter what. So these are probably rarer functions and things like that. In any case, if we feed in Rosetta structure predictions, David Jones's DMP fold structure predictions, or we feed in native structures, we actually get fairly similar performance, and all three of those origins for our structure predictions produce results that are dramatically better than if we were to predict on sequence alone. So this is, again, a bit of a complex figure, but the take-home message here is that if we have noise in our structures, we still tolerate that. The machine learning method has some ability to tolerate or denoise predictions. Okay, and I think in the interest of time, I'm going to skip through a few things here and get to why we call it DeepFRI. So the FRI stands for Functional Residue Identification. At least I think it does. Maybe the guys have changed what it stands for. We always try to give our software the strangest names possible, but in this case, this is a reference to the fact that when you fry food, it always makes it better. So, but it's also a proper acronym. Okay, so in the world of image machine learning, there's a lot of activity, and there's maybe a lesser amount in the world of parsing text and documents, but there's a lot of effort to not just predict, for example, that we have a dog in this image, but also to identify what part of the image has a dog. And so here what they're showing, what this sort of early paper is showing, is that if you have a prediction of a type of dog, you can then use various methods to go back through the model and find which pixels, if perturbed, would diminish this prediction. And so by sort of running backwards through the model, you can identify these particular regions as being responsible for the prediction of dog. And so now we get to the part that we're most excited about, which is that if we have protein structure as an input, and then we do things like salience mapping or class activation mapping, the question is, can we run the model in reverse, so to speak? It's not quite that simple, but can we identify not just the function prediction for a protein, but the actual residue resolution, the residue level regions in the protein that led to that positive prediction? And so Vlad has shown that works fairly well. And so if we look at this particular protein, which has a PDB, in that PDB are 2 calcium ions, right? And so this is not our prediction. Those 2 yellow balls are actually where those 2 particular very important ions are, right? So we know where they are. We also know from a database called BioLip, the residues that BioLip identifies as important for binding of calcium, okay, for chelating those 2 ions. If we run our model in reverse, we get this histogram from the class activation model, this histogram that I'm showing sort of at the bottom here. And the red areas in that histogram, these colors are mapped onto the structure. And so you can see that those red areas actually uniquely and correctly to what we know about the binding site. So in this particular case, the method has predicted the very small subset of residues that are important for that function. If we look at functions like DNA binding, of course, there are many examples in the protein database where we can very carefully benchmark whether we got the answer right. And so for this, the lac operon repressor, very famous protein, one of the first transcription factors characterized, we properly identify the DNA binding domain. For things with cofactors or substrates that are important, we also see that there are many crystal structures that include substrate, cofactor, various other binding events that we can use to benchmark this method, and it seems to work quite well. And again, this is the part that I'm excited about, so I have really just an insane number of slides about this. There's some cases still that are confusing or interesting. This is a case where we have glutathione. That's the function. It identifies these residues, but it actually doesn't identify this other domain. Maybe that's functional, maybe that's interesting, maybe that's just that machine learning needs enough to make a prediction and not more. So there's could be a lot of work there. So that's the end of this first section, and I want to show you just two more small things, just to let you know that even though I've taken a very deep dive into what we needed to do to get to this level of accurate site-specific function prediction, but if we can do this, right, we have a language model, right, as part of our method that's supposed to be capturing the hidden language of proteins. There's probably a lot we can do with that. We have methods for integrating structure and function. What if we tried to integrate network features? Once we have these predictions, what do we do with them? How do we share them with biologists? And so this is the classic situation in science where you solve a problem and the audience just looks at you and says, okay, now what, right? And so you always have to keep moving. So we solved this problem, and now we've enabled the study of other problems, and we've really upped the bar in terms of how complicated it is to share our data and our work with other people. So that's a good thing, but well, one of the key things with all of what I talked about still relies on alignment, right? We started the process with a multiple sequence alignment, and if you want to process millions and millions, eventually maybe a billion sequences, the process of finding your homologs is still something where we miss half the homologs. There have been many people who have talked or written about the problem that most homologous relationships in sequence are probably missed. So there's a sensitivity issue. There's also a speed issue. And so Jamie in the lab has been working on a new method called DeepBlast, and we hope to have a preprint out actually this week. So if you're interested in DeepBlast, we can share, send me an email and we can share that preprint with you the minute it goes up on bioRxiv. But Jamie Morton has built a way of using pre-trained language models to embed and then find relationships between sequences. And the really important thing here is that he's taken the language model, the embedding, the score for gaps, and the alignment method, and the entire thing he's made end-to-end differentiable. Once he did that, he was able to show that this is many thousands of times faster than BLAST. And in addition to that, it is dramatically more accurate, more sensitive than BLAST. And so here he's combining, he's comparing many different methods to the structure alignment gold standard that you get from these structure methods. And so the structure methods are showing us that there are, many more alignments that are detectable than what the sequences have. So the F1 scores for the sequence-based methods and many of the benchmarks we test, we can actually do dramatically better than BLAST and competing methods, but we still fall way short of what we can do if we have proper structure alignments to compare to. All right, the last little teeny piece, and then I'll save the rest of the time for questions, is something that we're working on broadly, which is protein design. And there are a couple of different approaches to protein design in the lab, and there are many approaches to the problem of protein design outside the lab. And I'm going to talk to you about a strange corner of the protein design world, which is designing small peptides and aiming those peptides at binding and interrupting or modulating the function of proteins. And so Vikram did his postdoc with David Baker and is now here at the Flatiron Institute in New York City and working as a fellow, which is a semi-independent position. In any case, he is a leader in the development of Rosetta for the purposes of design. And one of the key things that he's done over the years, along with Doug Renfrew, who's in my lab, and people in Brian Coleman's lab and Jens Meiler's lab at Vanderbilt, he's done a tremendous amount of work to not just model the peptides that are found in nature, but also to allow for the D and L amino acids to mix for side chains that don't exist in nature or don't usually exist in nature. So he's done a lot of work to enable those various things. And the key application that he cares about, and this is a slide from one of his recent reviews, is that we have this strange situation when we look at drugs. we have a large number of drugs that are small molecules, and here there's a handful shown. So acetaminophen, this is Benadryl, if you get allergic reaction, et cetera, et cetera. And then over here on the right, we have antibody-based drugs. And so these are either full-scale antibodies, which are many, not just proteins, but they're actually multi-subunit assemblages of proteins. Okay, and so whether we have fabs and fragments, camelid, doesn't matter, we still have whole proteins. Well, somewhere in the middle, we have a whole range of peptide, polykeep, all sorts of peptide and peptide-like drugs. And some of those are non-ribosomal peptides that include non-canonical amino acids that are found in bacteria. Some of those are drugs that we've recently made by synthesizing these peptides. And so this is where we want to aim. We want to aim at things that are roughly this size. And so this is a little tour of some of Vikram protein. This triangle shape here is actually a very interesting design that he's working on, where we have 3 peptides linked by this central molecule, which is just benzene with a handful of clickable groups. And so the peptides are synthesized, and then they're clicked onto this so that they have this tri-symmetry, this triangle symmetry. The triangle symmetry is really important because they're going after, in many cases, trisymmetric receptors like the receptor that's causing so much trouble on the surface of COVID. So this is something that lends itself towards the COVID-oriented drug discovery projects in the lab. And again, you can see here this Nature paper that Vikram published with his 30 closest friends, and some of the other pieces of work that he's put together recently. But the best is yet to come. Here's an example of using one of these peptides to bind and inhibit the beta-lactamase that is responsible for a lot of drug resistance right now. And of course, you can see that Vikram makes the most beautiful figures. The thing that is relevant for this talk is that when we predict that a given, when we design proteins and small peptides, one problem is we often find that a given conformation with a given set of side chains will do what we want it to do. But we have to make a lot of assumptions about how much the backbone will move in response to the changes we make in the design. And so what Vikram has found over the years is that if you make your designs, but you take your best designs and you put them through Rosetta and allow Rosetta to find the best 3-dimensional structure. If Rosetta agrees that the 3-dimensional structure, all the internal degrees of freedom of this peptide, if Rosetta finds that that's the lowest energy or the best structure it can predict, then you're much more likely to have a successful design. If Rosetta decides that it likes something different from the configuration you had in your design, then it's most of the time an error. And so forward folding takes many thousands of processor hours per molecule you want to test. And so Vikram has smartly encoded that process of forward folding into a neural network. And so the idea is, as the design process is running, this neural network can check each intermediate step in the Monte Carlo and tell you whether or not, if you were to forward fold that molecule, would it actually form that conformation? And so that's a very, very, very high bar to jump over. That's a very difficult thing. But what Vikram has found is that if he looks at the designs that pass his neural network figure, and he then looks at all the designs that were low energy, he finds that his neural network actually agrees with this forward folding more than it disagrees. And so if you look at things that have a good score with his neural network model, about 11% of them have good p-near forward folding scores. If you look at the population at large, it's roughly 5 times lower. And so this is still a work in progress. This is not something that is ready for prime time yet, but we have a lot of hope that acceleration of the protein folding problem and the protein design problem will go hand in hand, that some of the same methods will be useful in both cases. And again, one of the things that we're trying to do So Vikram and I agree that science fiction is worthwhile, and Dune by Frank Herbert is one of our favorite books. And so here he's shown the SARS-CoV-2 spike protein as a desert worm. It's trisymmetric, and so the idea is if we make a molecule that binds the closed form, we might interfere with SARS-CoV-2 spike glycoproteins binding the human receptors that it needs to bind in order to infect. And so we've made some molecules to that end. Vikram has got these molecules lined up. And so this is basically 3 cyclic molecules. And I hope that he's going to show this docked into the receptor. These 3 separate molecules then bind the closed form of ACE2 and keep it from opening to bind the human target. And so the machine learning method didn't produce this design, but what it did is it got rid of many, many thousands of designs that were very unlikely to fold. And so obviously we're all quite worried about this virus. Hopefully this works, but this is also something that is basic science in progress. All right, and so I again, put a bit too much in here. We had a deep dive into protein function prediction, and there I just wanted to show you some of the successes, but also some of the complexities and the future work that we need to think about to make function prediction better. We're very excited that we have site-specific function prediction and that some of those predictions look like they're working. Remember, we didn't tell the machine where the active site was. We just told it what functions many millions of proteins had, and it learned the active site de novo. So that's very exciting to us, even if it still is something that needs to be developed further. But overall, along with those other things I showed you, Vikram's work and Jamie's work, what I showed you essentially was a sampling of new methods for using machine learning to encode protein structure function relationships. And whether we use that to find new homologs through alignment, predict function, or design new functions, in the latent space, in the sort of the brain of the ML, there's lots of moving parts that we can reuse for different types of projects. So that's one thing that I just want to share. These different new methods enable all kinds of unexpected side tasks. And of course, we're not the only group exploring these relationships. There are hundreds of groups working on this throughout the world. So it's an exciting time to think about ML and its use to encode protein structure function. We can use this to scale things, right? So one of the nice things about the methods that we work on is that once we've encapsulated these things in machine learning, they're often much faster than the direct probabilistic MCMC modeling that we used to do. They're also really complementary, so we can scale and improve performance of protein design, but there are many methods that are hybrids of the two that we can think of in the future. Once we have these methods for function prediction, we have automatic site localization. So now if we think, does that enable us to actually do multifunctional design? And so one of the things that Vlad's working on now are methods to say, I want this function, but I also want it to bind this, and I want it to do that, and I want it to do this. So if we start to now think about multifunctional design, how does that work? How do we generate new sequences from the model that are likely to fold and carry out new types of functions and combinations thereof? So this is where we're at. This isn't the only project in the lab, but I thought that I would talk about these projects today because they intertwine to show us a little bit about the sort of intersection between what we used to know and what we model in biology and what we still don't understand in the machine learning methods that seem to be so powerful. And so this is hopefully a little window into the generative modeling meets ML side of approaching biology. And with that, I'll stop there and take questions. And thanks again for your for giving me the time to talk to you today.
So that was fascinating, Dr. Bonneau. There are many questions, and I would take some of them looking at our time. The first is by Dabulina. She is wanting to know what is the best way to incorporate network features or string for function prediction. She's wanting to know if there's any particular tool that you use or
yeah. So the person again, I'm going to chat his name again. So this is the Meet Bonneau is the person who is leading the charge there with Daniel, and the two of them are well, we have NetQuilt and we have Deep Enough, and these are methods, ways of extracting features from networks, including string. So you could use those codes already, and with those feature vectors, you could imagine just combining or pooling them with features from sequence. And so one of the things that we found in our early studies is that if you just take network features and sequence features and you're in a well-studied organism, the network features are much stronger at predicting function than the sequence features. And the reason is you're in a well-studied organism, but if you're predicting function, you're hiding functions, and you have to sort of make a proper test set. And so you have to simulate not knowing anything about the protein. And so if you don't know anything about the protein and its homologs, then you're building a situation that's a self-fulfilling prophecy that the network context will be more powerful than the sequence context. And so I'm answering this question in a very strange way, but one of the big problems when you integrate sequence and structure with networks is that it becomes quite complicated to figure out what's the right way to lay aside or set aside sequences for testing. You know, you have so much information there that you're sort of cheating on everything, right? Everything is already in the network or has a sequence or has a homolog. And so everybody that's function you know that you could use for testing is so integrated with your data structure that then it becomes a real challenge to figure out the fair way of testing it. But all of that said, NetQuilt is ready for action. And so that and Deep Enough can be used already. And if you search for Meet's name, then you'll find sort of his work with me and with Vlad, and that would be where I would point you now. That said, we hope that the best is yet to come, and the guys are trying to cook ways of getting the structure sequence network and the protein interaction network to live together. And we don't, again, we're having troubles now with the network features dominating that integration, but we've really only been working on that for a few months. So I have a lot of hope that some interesting things will come out there. So thanks, that's a great question.
So another is that in your experience between genes and proteins, what helps in tracing evolutionary relationships that are, had some experience?
Great question. Great question. So for very distant evolutionary relationships, structure information has given us a lot of really wonderful information. There are a bunch of sort of classic papers by a guy named Nick Grisham, yeah. And there he looks at sort of folds morphing from one fold into another piece by piece. And without sequence homology, because it's so distant, makes a lot of assertions about how different folds might actually have had their origin in each other. Of course, we don't know the directionality if it's loss or gain. But as you then transcend downwards, then sequence, protein sequence, I would imagine, is a place where we can be more sensitive in finding relationships. But the actual, fine grain evolution, the synteny, the position in the chromosome and the DNA sequence is important there. So I would say that, the answer to that question depends on what you want to do with that evolutionary information. If you want to understand structure function relationships and how they evolve, I'm partial, or I sort of think that we can get a lot from the protein sequence. But if you want to understand, the generation of new sequences and that sort of thing, then I think the DNA sequence is, of course, the place to look, because you can see quite a bit in the genomic context, and also the, you can see quite a bit at the fine scale.
So you're saying that the context of what one wants to look at is determined, it's genes.
I guess I'm saying for evolution, you need both. If you want to think of short timescale evolution, if you want to think of longer, billion-year arcs where folds are completely being neo-functionalized, if you want to do, if you want to think about it across many timescales, then you definitely need both pictures. Yeah.
So, because again, looking at the time, I will spend a little more time asking a couple of questions because they seem very interesting to me. So Shailesh is wanting to ask, what is the role of suboptimal folds in protein function prediction using ML algorithms? So,
Yeah, that's a great question. So suboptimal folds and suboptimal alignments, right? We don't always know if we've predicted something, whether it's right. The other thing, I suppose, is that could suboptimal folds teach us about the movements of proteins, right? So I've shown all these pictures. I didn't have any movies in my presentation, which helps with the bandwidth because we have to bounce all this off a satellite. But perhaps I should have movies because proteins do move. And so could we imagine suboptimal folds and things like that being ensembles that we can sample from when we look for allosteric interactions and the multiple functional states of dynamic proteins? I think that's really rich future work. I think that's a great question. Right now, we treat the contact maps and the proteins as static, but I think we shouldn't. And perhaps the fact that I don't have an answer to your question is just a reflection of the fact that the work is so new. It's all about a year old. But I think if someone wanted to study that, that would be a really interesting PhD, sampling from a bigger ensemble of suboptimal folds. That's a great question.
There we go. One more question. You spoke about QC mechanisms, and if you can just throw a little bit light on what kind of QC mechanisms are you using?
Yeah. So when it comes to the function prediction, we have multitask function prediction that includes the general terms and the more specific terms. And so when the function prediction algorithm is doing something wrong, what we often see are prediction patterns that don't reconcile with the tree in any smart way. So lots of very general, not so useful function predictions, and then a very strange smattering of more specific ones. So we often see, we can look at the way in which the gene ontology tree is organized to think a little bit about if our functions make sense. And some of that isn't quality control, but it's sort of filters for visualization and handing them off. When it comes to structure, we do actually have a lot of quality control there that we've built up over the years. The first piece of quality control is general to many different labs, which is that if you don't have a few homologs or a lot of homologs, then your contact prediction, whether you're using machine learning or coevolution, your contact prediction is probably poor. And if your contact prediction is poor, then you probably don't make very good structures. And so we need to have hundreds of homologs. Now those homologs don't have structures, right? We don't know the structure or function of any of the homologs, but having a view of what the variation is at the different positions in the sequence from the family of sequences, that enables a lot of the downstream work. So one of the first points of quality control is is the multiple sequence alignment of sufficient quality? The next key quality control point relates to if Rosetta produces the same answer more than once. And so if we find that Rosetta produces low energy structures and that many of the lowest energy structures have the same fold, then we're much more likely to have produced a correct answer. And so a sort of cluster tightness slash score is actually a reliable metric in many cases. Now we can tell when we got it right. If we don't see that clustering, it's likely that somewhere we got it right and we just don't know. The needle is still lost in the haystack. The grain of sand is still somewhere out in the ocean. So we often get correct answers and we just can't find them. And so that which also points to some really nice future work. But a very large fraction of the time these days we have good enough alignments and we can make predictions that converge. And so a convergence criteria is the main QC there. Yeah, okay. Great question.
So my next question is related to that actually. It is, so how does the structure predicted gets validated? Both, the validation of the structure as well as the function that is being predicted. What is the ultimate validation that you do for the same?
Yeah. Well, one answer to that is that we all of this code is still pretty new, and so the next time there is the critical assessment of function annotation, CAFA, will participate in that double-blind challenge. When it comes to the validation of the structure prediction, we're using methods that have already participated in CASP. So David Jones' method, DNP-fold, which is one of many really nice machine learning methods, and Rosetta, TR Rosetta is the TR Rosetta with content. It's a particular flavor of Rosetta that runs faster, but Rosetta in its many incarnations has been validated through CASP. The main way that we validate the structure function predictions are by hiding Go terms. We also have Go from 2016, 2018, and 2020. So we have some different snapshots of Go throughout the years. And so we can train with a very old version and then use the new versions to look at our predictions. And we call that a temporal holdout or a time holdout. Yeah, so there's a couple of ways we do that. The temporal holdout's our favorite for function. For structure prediction, we refer back to CASP because the people that run CASP are very meticulous in how they judge performance at both the global fold level, but also at even things like rotamer positions and things like that. So there's a lot of good work done on that we get to ride on.
Actually quite fascinating. So I'm taking my last question of the day, and the ones which are remaining, we will probably reach out to you and ask you to separately answer those.
Great, yeah.
But my last question for the day is actually, you can see it in the QA section. I'm just going to read it verbatim, is if we don't have a structure for the protein and we don't have high computer, systems, or what is the best way to predict the influence of nonsense mutations on the function of protein? Is there any application to study the effect of nonsense mutations? Understanding that the lab is not as highly equipped as one would expect. So in a setting, how?
Yeah, so we I'm going to just chat the name of this code because it's spelled strange, VIPUR. But we have in the past we've experimented with using Rosetta to interpret mutations, and mutations are tricky, right? Because when we try to model structure function, we do everything we can to look for distant homologs. So we're encouraging mutation, and then all of a sudden we get one really, and we're assuming that when we see sequences in other organisms that they are functional, but who knows, loss of function is a thing. And so we're taking snapshots of organisms, but many of the sequences that are in public databases, we assume that they're always functional versions, and that the mutations we see are those that are allowable by the thing that is, right? But some of those, who knows what percentage. If I quote this and we're recording, I'm sure I'll say the wrong number, but some percentage of mutations we see are non-functional. And so now we sit on a mutation, and we have to decide, does that mutation break something or not? And maybe we have a disease we're studying, and maybe we have a crop species that we're really interested in, but it matters. It matters whether that mutation hurts the protein or not. But because of all of our methods are trying to get as many homologs as possible, we have that conflict there, that balance. And so the first thing is just to say that's a tough problem, no matter how much computing or sequence data you have, right? It's a tough problem for everyone all the time. We looked a little bit at language models for interpreting protein function mutation. And I would say one interesting piece of work that you could look at, there's a guy at Facebook AI who actually, and his name is Alex Reeves, R-I-V-E-S. He actually did a lot of nice work with these language models. And again, these are models that you take all the sequences and you hide all the amino acids one at a time and try to make a model that can predict a hidden amino acid. He actually found a very nice ability to predict the effective mutations with that. And again, that one is just based on sequence. These language models are key components in how we predict structure-function relationships. but he found that by themselves they were reasonable at predicting protein function, protein function, loss of function mutations in proteins. And so he compared, he benchmarked the method looking at comprehensive mutagenesis experiments like those done by Doug Fowler in the University of Washington in Seattle. And so I think he actually benchmarked it fairly well, and it does look like that might be a good method if you want your prediction in a millisecond. You know, because whatever in machine learning methods, once they're trained, you spent some unimaginable amount of CPU to train them. But once Alex trained his model, now we get to use it, and running things through the model takes very, very short periods of time. So if you don't have huge computers, but you have some GPU somewhere, then you can run that. You could also probably just reach out to that group and they'd be able to do that. I don't know to what degree they've set that up as a server for making predictions at scale, but we could also run Viper as well.
So
it would take about a half an hour.
That's good. So Dr. Richard Bonneau, I can't thank you enough for spending your time. It's early morning there, and to get up, and the thing that you do is you start training and teaching people. But it really means a lot to us. It's the kind of time you've spent. We've learned, all of us here who are on the panel and all the participants. It has been a very good learning experience, and for us to know what's being done outside and where the world is reached. So thank you so much for spending your time with us. And there are certain questions which are unanswered. I would we would send it out to you and request you to answer if you can spend that kind of time.
Sure.
So thank you again. And before we close, I just wanted to make one announcement for the next webinar that we have, which is on 29th of October, is by Professor Rachel Natarajan from the Institute of Cancer Research, London. And the title of the webinar is Translating the Breast Cancer Genome. So we would look forward for all of you to join again. Again, thank you everyone, and have a good evening here and a good day to you.
All right. Thank you.
Bye-bye.
Bye-bye.